Okay, well, welcome everyone. We'll go ahead and get started. This afternoon, we're excited to have Dr. Brian Sturtevant with us. Dr. Brian Sturtevant, research landscape ecologist with the Northern Research Station of the U.S. Forest Service, working out of the Institute for Applied Ecosystem Studies, Lander, Wisconsin, for the past 17 years. He earned a BS in Natural Resource Management from Rutgers University in 1992, an MS in Wildlife Ecology from Utah State in 1996, and PhD in Ecology from the University of Maryland in 2001. His expertise is broad-scale interdisciplinary investigations of forest dynamics and disturbance ecology, with an emphasis on fire, insect disturbance, and the combination of field study, analyses of spatial data sets, and landscape simulation models. He also served on the advisory board and technical committee for the landscape disturbance and succession model, LANDIS-II. Let's welcome Dr. Sturtevant.
Hi, and thank you, Tony, and also Aaron, for inviting me here. This has been a great week, by the way. I had a great time working with colleagues here at the University of Maine, so I really appreciate the opportunity to come out. I'll speak to you today about some process-based modeling of spruce budworm dispersal and disturbance, so two different scales. And this is work that is funded in part by the Northeastern States Research Cooperative, as well as the U.S. Endowment for Forestry and Communities. That's part of the past Chief Tidwell's vision for kind of a one continent, one forest, one threat in terms of forest management issues that cross the international borders across the North American continent. So this is really joint work between the U.S. Forest Service and the Canadian Forest Service. So I was last here about 4 years ago at the Spruce Budworm Symposium 2014, and I was scratching my head at the time, like, okay, where was I at that time? And it's more like where were we, because I worked with a lot of other colleagues. But in essence, we had a spruce budworm atmospheric transport model that modeled dispersal, other related data. And we had plans for validation of that within Quebec. There was also the Border Lake study in Minnesota and Ontario with evidence for budworm forest feedbacks. And finally, we had an initial design for a budworm population model that's embedded within the LANDIS-II landscape model, with eventually plans for applications in. Let's see here. There we go. And so, first starting with a spruce budworm atmospheric transport model. This is a model that we published back in 2013. It's an individual-based model where we took insights and relationships from a landmark study in the air biology of spruce budworm published for New Brunswick into the last outbreak by Greenbank et al. And working with colleagues Gary Achtermeyer and G. Charney, the U.S. Forest Service, we developed this model that uses those parameters to define things like takeoff and ascent and horizontal dispersal and then descent. And interface that with weather inputs from a mesoscale model like the weather research and forecasting model. And that allows you to produce flight trajectories and deposition patterns based on weather. Since then, we've worked with colleagues, Jacques Lanier and Remy Saint-Amand, to embed this flight model into the biosim environment. For those of you not familiar with biosim, its initial purpose was to forecast the life stage phenology of forest insect pests or their host plants based on weather gradients. And so, in essence, what you're looking at is you take a whole series of weather station data, you overlay that with regional gradients such as topography that influence that weather, and just using the daily weather inputs, you can predict with really good confidence the different life stages of spruce budworm and other insects as it makes its way through the different larval instars, pupation, and then moth stage. And that's when the budworm flight model can then kick in. So once you get to that stage where moths are available, it will then interface with weather data and then produce your trajectories of dispersal patterns. And so if we look at that initial budworm flight model, we had some real simplifying assumptions within the model. And this would be an example of that. And so this would be some empirical data from that green bank study. And what you're looking at is the height above sea level in terms of aerial density of moths that was captured based on airborne radar data. And so we essentially took that distribution and embedded it within the model to define the height at which those moths level off. And you see the peak is about, or the mode is about 200 meters above the land surface. But we do know that moth flight altitude is dynamic. It changes from day to day, it changes from hour to hour, largely in part due to temperature gradients that it's responding to as it's flying above the inversion layer. And so we were fortunate enough to capture an exodus event from the north shore of the Gulf of St. Lawrence with a nice strategically located weather surveillance radar. And this is work done by, led by Jan Boulanger, the Canadian Forest Service. And from that, you can get at not only the directionality and the densities that you see, but also the height and altitude above the land surface. And so one of the key conclusions from that study is based on radar and lower tropospheric weather data, spruce budworm dispersed downwind in a rather shallow layer, somewhere between about 400 and 800 meters above the land surface. So that's quite different than 200 meters. And it's important because as you go up in altitude, your wind speeds change dramatically. And so that's going to determine where these moths can go, depending on where they settle out in that layer. So Jacques said, we can do better than that. We can base some of these on more biophysical principles rather than just using these empirical relationships. And so this would be an example of that. Based on the work of Deacon, you can base wingbeat frequency for your takeoff and sustained flight as a function of your mass and your wing area. That's m over a, with a constant that just needs to be estimated. And if you're able to do that, then you can determine the temperature at which moths should settle in the air mass, where their liftoff availability is equal to that temperature. And so the data from this, some of it came from canopy traps. This is work that was unpublished from Joanne DeLille of the Canadian Forest Service back in the late 80s and early 90s. And these are just canopy traps that are capturing moths as they emerge for flight. And looking at that, you can look at relationships between dry weight and forewing area and derive relationships for both males and females. And you'll see that the spread of the data for females is a lot larger than it is for males, and there's a good reason for that, and that's their egg loads. And so eggs weigh a lot, and so depending on how many eggs they're carrying determines that relationship. And so if you were to separate that out, spent females that have no eggs versus fully gravid females, you get a much tighter relationship. And that's important because depending on how heavy they are determines at what layer they end up flying in that surface as well. And so using that, you can estimate K by overlaying the distribution of those relationships with the wingbeat frequency that was estimated through radar data. And so this is the range of wingbeat frequencies that Greenbank et al. Had determined between about 25 and 42 hertz. And back calculating that, you see the liftoff wingbeats for gravid versus spent females. And this is important because if you remember that range, about 25 to 42, fully gravid females fall beyond that range. And this is pretty consistent with the biology of the insect. Fully gravid females are generally too heavy to fly. And so they generally will lay half their egg component before they do fly. There's also some other more indirect data to look at this relationship between wingbeat frequency and temperature. In this case, it would be the proportion of females that are taking off from some data published by Schaefer and Asoff, and also some laboratory experiments looking at male buzzing behaviors. They tend to buzz their wings before they take off, and they can determine that in the laboratory to look at that relationship between temperature and buzzing behavior. So from that, they're able to estimate these other parameters necessary for that. So that means that your altitude is now an emergent property of the temperatures that are observed at that time. You can do this for other traits. So this would be what Jacques calls the circadian rhythm of exodus flight. The timing at which the moths take off is also temperature dependent. And so when under warmer temperatures, they tend to take off later in the evening. Under cooler temperatures, they tend to take off earlier in the evening. And that can also be estimated as well. And so once you embed those kinds of relationships, this would be the overlay of the simulations of moth dispersal overlaid on top of that radar data that you see. And we get generally really good agreement between the radar data that we have and the moth dispersal vectors. And it's also consistent with observations of what we call ill-timed flights. These are pulses of trap captures that we see. And if you take a look here, you'll see if I can get this to work there. Yeah. Down here, there are some high trap captures located down in southern Quebec, as well as northwestern Maine and St. Field on this particular evening. And this is at a time when you don't expect local emergence based on the local phenology. There also wasn't any defoliation to indicate that kind of pulse. And so our trajectories are at least consistent with that as well. And that's about as far as we can take a qualitative assessment. You can look at the radar data, see if we get general agreement. You can look at these ill-timed flights, which in general it does a great job of doing so far. And so this would be an example for the past season. So looking at moth flights progressing over the course of the season. Some of the symbology changes through time as they updated their symbology. But in essence, what you're looking at is the where they're coming from, those pink dots. The brown is their trajectories, and blue would be when they descend prematurely due to temperature, whereas the yellow and would be when they're landing on land. Red would be when they land in the ocean. And so this gives a sense of what the dispersal looked like over the course of that season. And one of the, I think the take-home messages for all of you is that if you're wondering what kind of flights came from the north down into Maine, at least it doesn't look like there was much influx in this particular year. So that southerly flow of wind, which is observed sometimes, the most common is to the north or to the east. And that's in fact the direction that the outbreak is progressing. All right, there were some examples. I'll give you one. So right up here, he did record some flights that made it all the way to Newfoundland, about, I want to say July 30th. And our simulations are not showing that. So there's either something in our assumptions that are not allowing them to go that far, or there may be some specifics in the parameter estimations that we have that we're not quite getting them in the proper wind column or wherever they be. But there's a rich data resource available within this radar data that we haven't yet tapped into. And so we've now got processed data for the entire flight season of 2013, and I believe 2017 as well. And you can quantitatively compare the flight trajectories with the radar data directly to perhaps improve those parameters. But to do that, you need to do a few things. Number one, we know that the simulations are sensitive to the scale of the data that we put in. So data that you can download straight off the web is at a resolution of about 13 kilometers. And so that's missing some important things. We already know that. The other thing is that, and so the resolution of the radar data is about 1 kilometer. So ideally, we want 1 kilometer resolution weather data to match that. Number two, we're not talking about simulations of thousands of moths. We're talking about simulations of millions of moths in order to get at the proper density. And so that's going to require a whole lot more computed capacity. Well, we've got a postdoc, Matthew Garcia, who's working at the University of Wisconsin-Madison, who's doing just that. And so at this center, you've got 2 different options. You've got your high processing capacity computer, basically supercomputers that you can use to drive the high resolution weather models. And so those are already done now for the entire flight season for 2013. And that's because the processes within these weather models are interrelated. But in the case of your budworm flights, the moths don't care. They're not running into each other. They've got their own independent flights. And so those can be assigned independently according to these high throughput computing nodes. And so they have the software that simply distributes all of those across the different network within the system. And if there's not enough computing capacity within the system, it'll go beyond that to the open science grid. It'll then recompile all of those to get your produce these moth density grids across millions of simulations. And then you can compare that directly with the radar data to compute these accuracy metrics, essentially the same kind of methods that they apply to improve their rain forecasting, we can apply for improving the parameter estimations in the models. And so I'll just show you just a couple examples of that. So this would be the high resolution simulations. You're looking at the red trajectories from those overlaid over topography. And we're already getting some interesting features like interactions with topography right here, landings on the sites here as it makes its way through the valley bottoms. So some important features there. You can look at the individual trajectories of moths and also their altitudes as they're making their way through that system and the temperatures experienced by that moth as it makes its way through the system. And so then from that, they're in the middle of a quantitative parameter estimation as we speak right now, comparing it with that radar. So that is where that currently stands. So I'm going to move on now to some work that we've done in Minnesota and Ontario. This is work that we just recently published in ECOGRAPHY. Most of the work here you'll see comes from this paper, although there's some additional data on forest tent caterpillar that is currently in progress. But the study area for this is the Border Lakes landscape, and what makes this interesting from our perspective, if we're interested in whether or not there are important feedbacks between the budworm and the forest, this is an ideal location to look at that because you have very stark contrasts in forest conditions, both composition and configuration, because of very different land management legacies. So in Minnesota, you have a lot of harvesting that's going on in Minnesota. They've got a widely distributed road network that's essentially permanent and very fine scale harvesting going on that side of the border. If you were to go to the north side, they essentially build their roads in to get to the forest resources, and then they have much larger cut size in order to pull that wood out. So you just have a different scale of harvesting there, about at the same rate. And then in this middle area, you've got about 1 million hectares of wilderness between the Quetico and the Boundary Waters Canoe area that's essentially not harvested. So you have differences among those landscapes. And the way we approached this was through outbreak reconstruction using dendrochronology. This is the work of Louis-Étienne Robert. He's the PhD student with his that I co-advised along with Dan Nishaw at the University of Quebec at Montreal, and we looked at not only spruce budworm by analyzing white spruce cores, but also forest tent caterpillar by looking at aspen tree cores. And so here's the overall hypotheses that were driving this, that there's a number of ways that the forest can feed into the outbreak dynamic. The first is that it's essentially food. How much food is there? If you have a higher concentrations of food, that should influence cycle frequency and also the amplitude. And so that's going to feed into the dynamic. but you also have dispersal success. And so you have moths or you have larvae that are dispersing between trees. You have moths that are dispersing long distances. And depending on how much other forest is in the landscape, host is in the landscape, will determine the relative success of those, whether or not you're getting significant losses. And that can also contribute to cycle synchrony as well as the eruptive spread of the outbreak. And so in general, we're talking more host trees, higher amplitude, and more synchronous cycles. And to a large extent in the budworm system, this is what we see. And so what you're looking at here is a cluster analysis of those chronologies across these different sub-areas. This is the central area, which is essentially the wilderness area primarily. And you can see that, and also in Ontario, you get a pretty classic signature of budworm. About every 32 years or so is the frequency that you see. In some cases you get high amplitude, in other cases you get low. You could say there's an increase here, but we always hesitate to look at that for budworm when you only are looking at 3 cycles. Compare that with cluster 2 that is occurring down here in Minnesota. You get lower amplitude, higher frequency cycling that's going on in that area. Okay, so if you were to look now at the composition, so these are maps produced by colleagues at Iowa State and University of Wisconsin-Madison. What you're looking at here is a composite of tree species basal areas. So we've condensed it now down to host, which is shown in green, which is spruce and fir. And the blue is showing your non-host conifers, your pines primarily, but also cedar. And then deciduous species shown in red. And what we see in terms of synchrony, so this would be a it's a spatial non-covariance, non-parametric covariance function. Basically, you're looking at the degree of correlation as you increase in distance. And so these are pretty classic. You see a decline. Things that are closer to each other tend to be more related to each other. And you get this odd looking shape here in the wilderness. What this essentially means is that across the extent of the wilderness, things are essentially synchronized across that whole. If you compare that with Minnesota, you get a much steeper decline. And so things are far less synchronous in Minnesota than they are in the wilderness. In Ontario, it's kind of intermediate between the two. And so what's interesting about this is you know, take a look. This almost looks like you've got a bullseye here with a lot of hosts in the wilderness, which you'd expect in older forests. But if you look around the forest, there's a lot of deciduous that surrounds it, and that's what the forest tent caterpillar is responding to. And if you look at the patterns for forest tent caterpillar, you get the exact reverse. And so now you have higher synchrony in Minnesota and lower synchrony in the wilderness. And again, that Ontario managed areas are intermediate between the two. So now we've got 2 different species, each with different hosts, showing similar responses to the plants. There are some additional idiosyncrasies about this data. This, by the way, is Barry Cook. He's my partner in crime in this. He's a heck of a lot smarter than I when it comes to looking at this kind of data. But one of the things that he noticed in this data is that there's a lot of heterogeneity in cycle amplitude. So it's not as if this is a normal distribution. It's kind of a lumpy distribution. You tend to have kind of low intensity outbreaks and moderate outbreaks and severe outbreaks, especially when you separate it according to major outbreaks and minor outbreaks. Okay, so you're looking at kind of this would be essentially the harmonic of that initial outbreak pattern. And so we see that in the spruce budworm. We also see that similar pattern in the forest tent caterpillar. This is all the chronologies across the different sub areas. It's cycling a lot faster. That's what forest tent caterpillar does. But we get this kind of lumpy pattern in terms of its cycle amplitude. So why is this important? Well, if we look at one of the primary causes of synchrony, the Moran effect is essentially spatial synchronization via spatially correlated perturbations. So if you have independently oscillating populations, but you have a consistent perturbation that happens across large areas, that can then align those cycles. However, if the cycles are operating at different frequencies or at different amplitudes, that can complicate. That kind of heterogeneity that we see in time, we also see in space. And so this is just looking at these would be the primary outbreaks here, and the kind of the sub window here. So you kind of read it from here to here, here to here, and here to here. And if you look at this, often what happens is where you get a peak in one, you get a low in the next. And so you get this negative correlation across windows. So basically, they're out of synchrony with each other in space. You get a similar skipping behavior pattern that we see in forest tent caterpillar as well. So that kind of negative correlation, if you look between outbreaks, you get a similar pattern. So there's some interesting nuance that's going on that's not well recognized in the literature in terms of outbreak behavior. And so this brings us back to modeling their dynamics. And so if you look at the primary paradigms that drive budworm population dynamics, this may be review for all of you, I have no idea, but one of the primary paradigms is this multiple equilibrium paradigm. And so the thinking, this is championed by Holling and other folks like Ludwig et al. And essentially the primary driver is the forest. And so the cycle behavior that you see is due to the growth and decline of the forest as it's being attacked by the budworm. And it's the increase of forest that allows the budworm to escape that predator pit that changes the dynamic from an endemic to an epidemic situation. So that would be your kind of epidemics spread. And that now also has implications for your landscape dynamics as well. Dispersal is the primary driver behind the synchrony under this paradigm. And so this is where outbreaks start as an epicenter and then spread to adjacent areas due to the flow of insects. So the competing hypothesis was championed by Royama. There's harmonic oscillation, which is that delayed density-dependent response between your predators and its prey, the budworm. And so really the emphasis there is between these two components here, and the landscape implications of that is that the Moran effect can have a really powerful influence on that because it's the same oscillation that it's aligning. Okay. So one of the reasons why this is so hotly debated, we argue, is because the budworm system has elements of all of these. And so we believe that a hybrid of these two paradigms is actually what's happening with it. And so that's what we're attempting to model. And then finally, all of this falls within the realm of climate. So when you think about your regional dynamics, there's that sweet spot in terms of where it's thermally best for this budworm to operate. And that's where you see the most critical outbreak dynamics occur. And so the model that we're using to look at this is the LANDIS-II model. It's a forest landscape simulator. It simulates tree species cohort biomass on individual cells. It has seed dispersal that influences the spatial pattern and interactions among species. And then the disturbances, you can have insects, harvest, fire, wind. We've had browse disturbance that we've recently added, any number of different disturbance extensions that overlap in space and time. So the budworm disturbance extensions, the team that's working on that, myself, Barry Cook, Brian Miranda, U.S. Forest Service, as well as Dina Thapa out of Iowa State University. And this is kind of an overview of that, but I'm going to walk you through the different components of the model. And so essentially at its core, it's based on the principles from Royama, these conditional recruitment functions that drove that predator-prey dynamic. That's what's driving the model. This is straight out of his book. And so essentially what you're looking at is the effects of natural enemies on each other, crowding effects. You've got the effects of budworm on each other, crowding effects for them, and then you've got the interactions among those. So you've got budworm on the predators and predators on the budworm. And those nonlinear responses are what result in those outbreak dynamics. And so using those parameters, we can generate outbreak behavior that has different characteristics. And so, for example, you can look at this kind of more smoother outbreak, more harmonic oscillation outbreak, and flip between kind of a 35-year cycle and a 24-year cycle. These are cycles that are commonly observed across the range of the budworm. And you can do that by just simply adjusting one of the parameters that represents, in this case, parasite fecundity. If you adjust things like crowding effects for budworm, you get this more eruptive behavior. So much higher amplitude. You've got a much stronger kind of overreach of the simulations as well. And so that's how that would respond to that. And there are other ways of kind of adjusting the state space. What our thinking here is that if we can find the right state space and then apply that to our landscapes, we can generate similar looking outbreak simulations. So that was the predator-prey, but where's the tritrophic element of that? So tritrophic means predator, prey, host. And so this is where that's inserted. This actually is adapted from work that Renier and you had done in 1991. It's estimating defoliation from L2 larvae densities. And if you can do that, you can then feed that information back into larval survival, and that is the key feedback so that you get the response of declining food on the budworm population. And then in terms of the impacts, the response of the defoliation on the trees themselves, that's been derived elsewhere. This is work done by McLean and others on the spruce budworm decision support system. It's just simply a lookup table of your stand impact matrix. So it's the cumulative defoliation will have host specific responses, and that will be reflected in both growth, impacts, and mortality. This was adapted for biomass succession by Chris Bennett. So those have all been embedded. The first model you looked at was a simple Excel spreadsheet. Now we're increasing complexity. What we're looking at here is a simplified version of LANDIS-II, where you're just looking at what's occurring on an individual cell. But now we can include the feedbacks of the host in the equation. And so just to show you a bit of what that looks like, if you run just balsam fir by itself, you get kind of a classic wave dynamics in balsam fir. This is replicated, which is why you see the variability in the curves there. But if you add budworm impacts to that, you get that sharp decline, and you can see those peaks that are, and peaks and troughs that are related to the impacts from the budworm outbreaks themselves. You get a much more, if it just happened to be budworm or balsam fir by itself, which of course never happened, this might be the pattern. And on top of all that, you still get the same population cycling that we parameterized in the Excel spreadsheet you get out when you look at it from the individual site scale. So now let's add space. Okay, there's a number of ways that space contributes. And so there's local dispersal through the larvae. This was basically a neighborhood average around an individual cell. And then you've got two different methods of dispersal in the adults. You've got short distance dispersal. This is what budworm ecologists often refer to as gray to green. And so they go from locations that are being heavily defoliated locally to places that have more food availability. So this is being weighted by host composition. Then you've got your long distance dispersal. This is on the order of a kilometer. This is on the order of, tens to hundreds of kilometers. It can be, number one, it's conditional on host damage, so it's not occurring unless there's active host damage on site, and it can be directional. Finally, you've got natural enemy dispersal. So the natural enemies have to move as well. And we had no idea how important this parameter would be. It turns out to be really important, which is good news, bad news. It's great for theory. It's bad because it's poorly measured, right? The other is, of course, the Moran effect. So you can do a perturbation. In this case, we just did it as simply as possible. Larval budworm survival, that's spatially autocorrelated. Eventually, when we get to this kind of scale, we can include things like regional dynamics. This is the population growth index that was calculated by Bernier et al. And this shows that sweet spot that where you have the highest growth rates of budworm, depending on the kind of southern constraints and northern constraints. This is one of the things that will allow us to pick it up and move the model elsewhere. And it's also a way to address questions like climate change. So let's see what some of this looks like. This would be simple test landscapes that we started with. These are 10 by 10, so it's 10 by 10 miles in size. The cell size is a hectare. And it's just simply initialized with homogeneous white spruce. Okay, and so what you're looking at is, it starts out as being synchronized outbreaks. This is defoliation in terms of percent. And you'll see that it quickly breaks down. And so if anyone would like to have a lava lamp looking screensaver, I have a lot of these. Okay, and this is what happens essentially if the natural enemies have a hard time keeping up with the budworm. You get traveling wave dynamics, and that completely obliterates that initial outbreak frequency that we saw, that we were putting in. And if you were to look at that, so the way to read this graph is you're looking at white spruce biomass up here. The colors are individual cells, what's happening on an individual cell. The dashed line would be the landscape average for that. You can see how the host foliage is responding. You can see that, you know, when you initialize things homogeneously, it holds on to that for a little while, but eventually it devolves into that spatial asynchrony, okay? And so you get, there's a number of things that happen when it gets into that state. One of those is it decreases in amplitude, especially across the landscape. It also increases frequency. Okay. These are just some diagnostics that we use, and I don't think, and I'm not going to ask you to memorize what all of these are, but essentially what you're looking at here, host biomass, what's happening to the foliage, and this is the percent defoliation over a 1,000-year simulation. So we're kind of looking at what happens over the long term in the system. We can use things like periodograms to look at the frequencies in terms of their outbreak dynamics. And so essentially what you see is that breakdown that I showed you earlier. Okay? One change to that same parameterization, you increase natural enemy dispersal to about 2 kilometers, and now that enables you to have a fully synchronized outbreak, at least at the scale of a 10-kilometer or 10-mile square landscape. And so we know that if we were to increase the scale beyond that, it would still be sensitive to this parameter. So this is something that is a real issue for us as we move forward. We've done hundreds of different experiments, but this would be an example of one of those where we're looking at the effect of different host patterns on the dynamics. And so you can have different proportions of host versus non-host. Essentially what we're doing is we're initializing the green and white spruce and the yellow in aspen. And you can have them in different configurations, so either highly clumped or randomly dispersed. And so just to show you, this would be this this initialization right here. So about 25% host, coarsely represented. And what you'll see is it starts out as synchronized, is what it normally does, and then it breaks down. But at some point, it tries to synchronize, falls apart, and then synchronizes. So what do you suppose allows it to synchronize? Well, if you look at the trend in host biomass, that is trending upward because the white spruce is eventually gaining more ground. And as soon as it gets above a certain threshold, that's what allows it to remain synchronized at the landscape scale. And to see what that looks like, you can see that here. Okay, so all you're seeing defoliated is just where the white spruce is located. You'll see the white spruce beginning to gain ground in terms of initializing underneath the aspen that's adjacent to it. It'll break down into that asynchronous behavior. And as it's increasing in biomass, it's trying to synchronize. And eventually it gets to that fully synchronized as it increases above a certain threshold. So where are we going with this? We've got some, I think, a useful model. It's still in the early stages of application. We're still using simplified landscape. When we apply this to realistic composition, we're not getting the same dynamics, so we're working towards that. We're working towards scaling this up to the scale of the border rays. But one of our applications will be an initial validation within the state of Maine. And so what we're doing, we're working here with Casey Lagarde and Aaron Simons-Lagarde on this part of the study. And the question is, how does the model behave when applied to the forest conditions that were representative of conditions before the last major outbreak? We would expect to see highly synchronized, high amplitude behavior. But to do that, what Casey is working on is producing what the conditions were about that time period. So he's doing some hindcast mapping of spruce fir and other tree species at that time. So that's a work in progress. And so let me just sum up here in terms of the spruce budworm disturbance extension. We've got a lot of good that's coming out of this. We get emergent outbreak behavior based on close to first principles. Budworm is responsive to the landscape host. That's what we like to see. It uses these empirically based impact functions that we believe have good, strong grounding in what actually happens in terms of the response to the trees. We get interrelated outbreak characteristics. So synchrony is related to frequency, is related to amplitude. That is precisely what we see in the border lakes, as well as that spatial behavior. We also see similar things in our own simulation. And then, again, the results for these simplified landscapes are consistent with the outbreak dynamics that we observed not only in the border lakes, but in other places in this range as well. The bad, we still have work to do. Among those is getting it to have similar behaviors with more realistic full species composition. We're not there yet. This I see as a calibration issue. So we'll work through those to see if we can get our outbreak dynamics to behave once you add the full component of species. We're working on adding directional moth dispersal. So right now it's just simple isotropic dispersal, but if you think about it at the scale of 10 by 10 square miles, when they're dispersing hundreds of kilometers, essentially what we're doing is we're wrapping them around, and so they're going to land in the landscape at some point. And so because of that, we'll also have to address, when we get to real landscapes, the issue of edge effects. They're very sensitive to edge effects, which is why we wrap them in the first place. But once we get to real landscapes, there are real issues with wrapping dispersal. And so we're going to have to bridge that as well. And then finally, including the climate factors, we have a method to do that. That will increase affordability, but that's all work to come. The ugly. Spatial nonlinear models are complicated. There's just no way around that. That's and like I said, if anyone would like a screensaver, we have lots of them. One of the outcomes, it's ugly, yes, but it's important, and that is that ability of natural enemies to track budworm in space is a key element. That I don't think has been widely appreciated, so it's something that we're interested in investigating further. And I think I need to emphasize this will never be a forecasting tool. So we should never look at it as if we can apply it to the current landscape and predict what's going to happen over the next couple of decades. That's not an appropriate application of the model. But what we hope to do is project the bounds of system behavior. What kind of outbreak behavior might you see both now and based on the kinds of activities that you're doing now, what might you expect in the future? that would be a much more appropriate application. And with that, I want to thank all the supporting agencies and funding agencies that supported this work. And I'd love to take any questions you might have. Yes.
So, did know you other radar?
Yes.
Do you have an outreach component of your work, or when you like your app on your?
No, we don't. You know, the Canadians. I mean, that's where a lot of this is happening, and it's especially now that moths are raining down in more populated areas of New Brunswick. It's all over social media. That's happening everywhere. People are taking photos of swarms around, you know, car dealerships and all other stuff as they coat the ground. It's amazing. And so there is an outreach component. And the maps that you saw, those have been distributed to a number of people, I think, in this room, as well as others that have interest in this. And I know certainly, I think the Quebec media has also been receiving some of those maps, so that when those kind of rains of moths occur, they know that we're actively looking at that. So there is a component, but in terms of the radar specifically, no, but it's pretty impressive when you look at the moth densities that come out of that. Oh, there you go. There you go. Right on. Yeah, that's excellent. And I know that I've been very interested in connecting with the National Weather Service folks up in Caribou. I think they actually have better radar. It's just that they don't have enough moths floating past their radar yet to make it worthwhile. So there's a number of layers that are happening at once. I do think that this kind of skipping behavior has been documented before in other places, where you have alternating high intensity outbreaks in one location. The next outbreak tends to be. And so outbreak hit New Brunswick really hard last time and may not hit it as hard this time. It didn't hit the North Shore very hard last time. It's certainly hitting it very hard this time. That may be climate related as well. But again, these are layers of things that are happening at the same time. So that's number one. Number two, they've learned a lot about the early stage of the budworm increase that was underappreciated in the last number of outbreaks. And so this early intervention strategy, I don't think there's any doubt that it's working. If you look at aerial surveys, essentially the defoliation stops at the border. It's impressive. So it does appear to be working. So you've got that going on. Forecasting how precisely that's going to work out is, I honestly, I would have a hard time forecasting that. I think it is a matter of time before it hits Maine, but I would suspect the impacts are not going to be like what you before. And I guess that's what I would suggest. My gut reaction, looking at what we see at the Border Lakes, what we've seen elsewhere, is that I would expect a light outbreak. You know, that I don't know that we even have the capacity in a model like this to be able to evaluate that. I just don't know. It's a good question whether we could apply it in that sense. I mean, technically, we should be able to. We can knock down early populations. There would be a way of doing it. I really don't know what the outcome would be. It's a very good question, but it's a hard question to answer. Now, Jacques Grenier, he might have a stronger opinion on that. berry cook may also have a stronger opinion on that. So, you know, I imagine that that's of real interest here, and I wouldn't mind taking that on. I'm just not sure I'm the right person to answer that. Start a hedge. Yes?
If you look at sensitivity to differences in host susceptibility, so fur versus
We have, yeah, and so we haven't yet gone to black spruce. And black spruce in general we consider call a spillover species just because of its asynchrony with bud emergence. And so, but it does absolutely, if there's mixtures of black spruce and fir, larvae will find it and will defoliate it. So that's really, I think those are fairly straightforward things. We have looked at balsam fir. One of the reasons why we started with white spruce is just that it persists longer. So if you want to have long simulations, you don't want to kill off all your forest. You start out with something like white spruce that can persist longer. So that's why we started with it, to understand the gross behavior before adding in things like total decimation of the forest. But we have done simulations with just balsam fir, for example, and it does behave somewhat different.
Any other questions, Brian? Well, we're going to reconvene at Tim's tap room, so please join us for that. Thanks, Brian.
Thank you.
Good job. Thank you.
Thank you very much. Yeah, I'll have you.
Yeah, I'll just stop this.